I'm going to demonstrate the electron transport chain in photosynthesis, which is very similar to the previous lesson, which took place in the mitochondria. Now again, like I did with the mitochondria, I'm only focusing in on the membrane where the reactions are taking place. And again, the students can conduct a series of before, during, and after diagrams, and they can use words to explain the pictures of those before, during, and after diagrams. So you have your legend, so you can see that the electron transport chain is set up, and I have a water molecule here. Now to start this lesson, I put a pair of electrons in photosystem 2. So you can sketch a photon initiating this reaction and bumping the electrons from the protein onto PQ, the shuttle. And that's usually the way I'll start. Then I'll go to the photolysis reaction. Now, one thing I've done with just using paper is, after I conduct the reaction, but I can show you this now, is to split the word because we're splitting the water molecule. And just an interesting thing that I focus in on at this time is, even when I split this, okay, photo means light, lysis means to split. That still is not specific to actually what happened, right? The students just can't write, you know, okay, in light, something is split, which is what that language is. They need, in the presence of light, a protein is splitting water. So this helps them kind of remember what photolysis is, but they need to add a layer onto that note. So again, as in the mitochondria, before I do the reaction, there's the proton, right? I've placed the water molecule on this transparency, and you can see that kind of spin move that I can do. And that's just for, well, A, it's fun, and B, you can speed that water molecule up as fast as you want to show the speed of water. So this photosystem conducts that reaction where, when the reaction is over, I'm going to just pull this down a bit. The way I'd like to do it is, we've got protons, electrons, and oxygen. So there's our after picture, right? And I just leave that for whatever amount of time it takes for the students to absorb. And 99% of the time, if not all the time, they want me to reverse that. Sir, show us that again, right? And they're doing their before, during, and after diagrams. The lost electrons are replaced, right? And these protons are going to start to diffuse over to ATP synthase, which we'll get to in a minute. I tell my students, because again, they're really concerned about this oxygen, which is a good thing, but we'll come back to that oxygen in a bit. So we just continue to follow this pair of electrons. It's going to, that FQ is going to pick up that proton, right, from the stroma and deliver to the B6F complex the electrons and that proton. The B6F complex pumps that proton into the lumen. The next shuttle picks up the electrons and shuttles them off to photosystem 1. Now, the electrons in photosystem 1 are not present. So let me just back this up at this point. So you only have 4 electrons in this model. So what I will do here is, what I'd suggest to you is to take these electrons and place them onto photosystem 1, and to communicate to the students that both of these photosystems, for the reasons of whatever those wavelengths of lights are that you're going to teach, they're both simultaneously being hit. They're both liberating those electrons. So I've demonstrated the initial, I just started the reaction here. Obviously it's happening everywhere, and that's why I'm moving these electrons now, to show the students at the same time this photosystem loses a pair of electrons when that photon hits it, and those are replaced by the shuttle. And these electrons are shuttled over, picked up by NADP reductase. Notice we're going to carry out our last redox reaction here with the coenzyme NADP. And again, you'll notice that there's a red background to it, which is representing that there's ribose in there. So we refer to that as an RNA molecule. And the P is colored green because that's a phosphate. And in our model, like our phospholipid head is green because it contains that phosphate. So there's that color theme here of an RNA molecule with that phosphate. So the coenzyme NADP is going to receive those 2 electrons. It's going to pick up a proton. And to get ready for this particular reaction, I'm holding in my hand the hydrogen already, so I can change that to, now it's NADPH. And that's going to move off into stroma for the exciting Calvin cycle. Now, the only other thing we need to address really is we had this water photolysis happen, and I only modeled 1, and we're left with this free radical. So I would what I normally do is I don't conduct photolysis all again. I'll just build a water just with the hydrogens and oxygens and tell the students that simultaneously another water molecule goes through photolysis. And here they see now the complete reverse of cellular respiration in terms of the relationship between water and oxygen. So in photosynthesis, water is split, and that's where the oxygen comes from that we end up breathing, and the reverse happened in the mitochondria. So that concludes my demonstration on the electron transport chain in a chloroplast. Enjoy yourselves with it.